unit. And they're out of order. If you've been paying attention and that bothers you, I'm sorry. Um, there's different philosophies. The College Board has it in a set way. Um, and some of the philosophy behind doing this one, this is unit 6A, and we have not done 5 yet. So we finished up with 4A and we finished up with 4B. We did immune system, we did homeostasis, we did cell communication, we did the cell cycle. But then the next unit, according to College Board, would be to do meiosis and genetics. And I really wanted to do that because I know a lot of you like genetics, and that's kind of a nice, easy start to the next semester. However, the next unit after that, 6, we've split into 2, 6A and 6B. And 6A is what we're going to do first before we do 5. And the reason we're going to do that is because we're going to talk about DNA and we're going to talk about RNA and their structure and how you make a copy of a DNA molecule. So when we were talking about mitosis, we talked about how during S, we made copies of DNA. Now we're going to actually talk about the enzymes that are responsible for making DNA and how that happens. If we go straight from 6A to 6B, it gets a little bit dicey because 6B is complicated. It's gene expression or protein synthesis, and it's difficult. So I like to do 6A first because it's not easy, but it's short. And then we go into genetics after that. So after we talk about how we make these chromosomes, now we talk about, in genetics, we talk about how those chromosomes are passed on from generation to generation. And then we'll go into gene expression, which is now that we've combined our genes and genetics, mom's DNA and dad's DNA. Now we can talk about how those genes get expressed in genetic gene expression. So it flows one way or the other, depending on how you want to do it. And like I said, I went back and forth, and I decided to do it this way because I think this will be a little bit easier on you. So only one chapter in this unit. Test is going to be a week from Friday because it's short. Watson and Crick are credited with the structure of what DNA looks like. That was very important for all of our later tech that we now kind of take for granted. But back in the day, we didn't know that. Watson and Crick introduced an elegant model called the double helix to tell us what DNA, what the structure of DNA was. Back in the 50s, they didn't actually even know that DNA was the hereditary molecule that got passed down from parents to offspring. So if you looked at an early biology textbook, it didn't exist. They had some suggestions, but they weren't really sure what it was. Turns out it's DNA. We'll talk a little bit about that history before we get into the nitty-gritty of what DNA is made of, and then how during mitosis, during cell cycle, or I should say during cell cycle, before mitosis, we make a copy of our DNA. We'll talk about that. But first, a little bit of history. And it's pretty brief. We won't do too much of it. Again, before we even knew that DNA was the hereditary molecule, there was a gentleman by the name of Erwin Chargaff. You won't need to remember names. Who was studying DNA. And he found that, what did he find here? That wasn't what he found. Yeah, okay. So we already know this. So we're kind of going back in retrospect here. We know that DNA is made of nucleotides, right? Remember what the nucleotides are made of? Nitrogen base, sugar, and a phosphate. So sugar, a phosphate, and a nitrogen base. And those nitrogen bases could be adenine, thymine, guanine, or cytosine if you're talking about a DNA molecule. They're slightly different in RNA. What Chargaff was looking at was the different composition of the nitrogen bases, because that's the only thing that's different in DNA molecules between people, or between people and rabbits, or between people, squirrels and, donkeys or whatever. So he was looking at that composition. And his big finding was what we now call Chargaff's rules, which was, and this is really the key point here, is that it didn't matter what species you were looking at. The number of adenines in a DNA molecule always equaled the number of thymine molecules. And the number of cytosine molecules always equaled the number of guanine molecules. At this point, they didn't really understand why that was. But they knew that if they had this much cytosine, they were going to find this much guanine. Right? If it's 20% guanine, it was 20% cytosine. If it's 50%, that would be weird. If it was 40% adenine, it would be 40% thymine. Okay, it always matched. That's not very clear, is it? I don't know why it's so fuzzy. But again, we've got this sugar phosphate backbone, and I'm showing you stuff that we know they didn't know back then. Okay, they didn't know. They could take this stuff out of a nucleus. They could look at it under a microscope, but they didn't really know how it was all put together. And we know that we've got a phosphate, different experiments, and we'll talk about some of them. So we've got a phosphate followed by sugar, followed by another phosphate, followed by another sugar, phosphate, sugar, and it goes on. And each one of those sugars is attached to a nitrogenous, not a nitrogenous A. What am I thinking here? A nitrogenous base, either thymine, guanine, adenine, or cytosine. After DNA had been accepted as the genetic material, and there was a series of experiments on that, because we didn't know back in the day that DNA was actually the molecule. But once they understood that it was a molecule, then it became, okay, well, if DNA is what's passed down from generation to generation, we need to know more about it, because maybe some of the diseases that we have, maybe we could alter those diseases. Maybe we could keep those from happening. This was, early 50s, but maybe we could keep those from happening if we could alter that genetic information that was passed down. So you wouldn't have different genetic disorders. And that is, even just today in 2024, just becoming, we're starting to see that, where we now have some techniques where we can actually we can edit the DNA of people and get to the point where we may be able to get rid of some diseases, hopefully all diseases at some point. But again, that all started with, what exactly is the structure of DNA? So I feel sorry for this woman, because I feel like this is the only picture that was ever even shown of her. This is Rosalind Franklin. And Rosalind Franklin is noted for a couple reasons. One is that she took the first pictures of DNA. And that led to Watson and Crick, who put it all together, led to them being able to do that because of those X-ray pictures that she took. So Franklin, in her, she had crystallized DNA molecules, and she could use X-rays to take pictures of them. And from that, she was able to figure out how wide the DNA helix was, and the spacing of the nitrogen bases between each other. And that was a really key thing, because what it did was it told researchers that DNA was a double helix. And they didn't know that before. So we know now, again, it's kind of weird. It's like, well, duh, we know this. But they didn't know it back then. We know that you've got this sugar phosphate backbone. And then you've got these bases in between. And so Chargaff said, well, A always paired with T, and it's like, and C always paired with G. And Rosalind Franklin says, well, and the diameter has got to be this far apart. So they kind of pieced everything together from these bits of information. And before I go any further, I just want to talk a little bit more about Franklin, because it was actually both. I skipped a slide. It was both Franklin and Wilkins that determined this. And you can get into a whole history of it if you desire. There's autobiographies. There's been biographies written about Franklin, and there's been an autobiography written by Crick, Francis Crick. But I mean, it goes to the politics of science and just of culture in general, because this was in the 1950s, and she was a woman in science, and this was the 1950s, where women traditionally stayed at home and raised the children, and they did not have a career. And so Franklin had a lot of issues with that, because people were like, why aren't you at home? Why are you here? Why are you? She didn't have, when they had like a smoking lounge, believe it or not, where you could just go and smoke to hang out, where all the scientists would go and hang out, except if you're a female, you weren't allowed. So she smoked in the bathroom, okay, because she didn't have a place to hang out. Crick and Watson took her information. They took it before. She didn't give it. They took it.
I was going to ask about that.
Wilkins actually gave it to them without Franklin's permission. So it was kind of a big deal. And Watson and Crick won the Nobel Prize for their work. And even worse, I mean, this was years later. Nobel Prizes aren't given like the next year, because it takes a while for people to understand how important research is, a particular kind of research is. So it took a while for everyone to realize that this DNA structure stuff was really important. So I don't know, it was 10, 15 years later. It was later. And Franklin would have probably taken part in that Nobel Prize, but they don't give the Nobel Prize posthumously. You know what that means? After you die. So she was already dead by that point. You know why she had died? Anyone study this? Cancer. You know where she got cancer from?
Smoking.
No, actually, she didn't. She got cancer from the X-rays. They didn't know back then that X-rays caused cancer. So she was doing all this radioactive studies without protecting herself because she didn't know it. Yeah. Well, I mean, she's gotten recognition now, but she never. They still don't give the Nobel Prize posthumously. I think part of it is you get like $1 million when you win the Nobel Prize. So I would think they would give it to their family, but that's just not something they do. Anyway, that is the story of Rosalind Franklin, and there's an autobiography that I tried to read. No, not an autobiography, a biography I tried to read. It was pretty dry. And then there's about Franklin. And then there's another one by Crick, who last I heard was still alive. But
What?
How old is he?
Crick was it Crick or was it Watson? I think it's Watson. I can't remember. One of them was still alive. One of them was the mentor, and the other one was the grad student. But I don't want to talk bad about anybody until I figure out who's who, because one of them was, well, kind of a jerk. Yeah, DNA history. So it's a little drama, and things like this do happen in science. You do see that sometimes scientists are very cagey about their research and don't want to share it until it's been published, because they are worried about not getting the credit. And it's not a thing about ego, it's a thing about job security. So if you don't publish enough papers, a university may say, well, you're not publishing anything. You're not bringing any money into the university. And because you're not bringing any money into the university, we're going to let you go. Okay, so it's not just about prestige; it's also about being able to keep your job. And so a lot of professors, I know my experience. My professor was he did not want to share his information until it was published. And that's kind of what got me out of research, was the fact that no one was sharing anything, and science moves very, very slowly because of that and some other reasons as well. So anyway, lots of drama. So Watson and Crick built models of the DNA double helix that conformed to the X-rays that Franklin did and the chemistry that Chargaff said. Okay, A's pairs with T's, and it's got to be this wide. Franklin concluded that there were 2 outer sugar phosphate backbones. Okay, we know this already, but they didn't know it back then. Sugar phosphate, and that the interior of the molecule was nitrogenous bases. If I brought this one out, probably have already brought it out. I brought this out before, haven't I?
No.
No. Yeah.
Yeah, you have.
Yeah. So this is DNA. If I unwind it, the black and whites represent the phosphates and sugars. The sugars are the whites, and the sugars attached to the nitrogen bases that are the 4 different colors, representing adenine and cytosine and guanine and thymine. Okay. So Watson built the model in which the backbones were anti-parallel. Do you remember anti-parallel? Yeah, they run in opposite directions. Good. You've got one going from 3 to 5 direction, and the other one flipped 180 degrees, going from the 5 to 3 prime direction. Did I say that right? 3 to 5 and 5 to 3. At first, Watson and Crick thought the bases were paired like with like, so adenine would pair up with adenine, thymine with thymine, and etc. But after looking at Chargaff's results and also looking at the X-ray diffractions that Franklin did, they realized that no, it had to be a smaller molecule pairing with a bigger molecule. So if you remember back in the day, and that would be all the way back to last year, we talked about how we had adenine and guanine, and we had all things that are good are pure. Remember that? All good, pure. Those are purines. And those purines are 2 molecules, or 2 rings, I should say, 2 rings wide. Okay, so purine is a small name, but it's a bigger structure. And then King Tut lived in a pyramid. So that's your thymine, uracil, and cytosine. And they have a smaller ring structure, those little guys. So if you had 2 little guys, so if you had a thymine and a thymine, that wouldn't work, because if this was a T and a T, that wouldn't work because it had to be this distance. But if it's that distance, then these molecules, there was too much space here. So that wouldn't work. And if you did, say you did adenine with adenine, they were too wide. They kind of bumped out here beyond the distance, beyond the width. So that couldn't be. But if you'd use an adenine with a thymine, it was just the right distance. What's represented in the middle that I'm not showing you here? Oh, that wasn't a very good. I think I heard it. Yeah, there's H bonds there. That's representing the space that's in between them. Okay, so hydrogen bonds hold the nitrogens and phosphates. Nitrogens and nitrogens together, the A's and T's and C's and G's. Okay. Okay, so Watson and Crick reasoned that the pairing was more specific, dictated by base structures. They determined that adenine only paired with thymine and guanine only paired with cytosine. And that then would explain why Chargaff always found that however many guanines you always had the same number of cytosines, because they were paired together. They were a matched set. If you didn't have one, you didn't have the other. Okay, adenine with thymine and guanine with cytosine.
How did they figure out like the size of each
that was based on the X-ray stuff. So when they looked at them, they took pictures of it. And don't ask me how, because I'm not exactly sure. But when they looked at it, I mean, you can see by going up here. You could see how wide it was. And because it was so wide, you knew that. You know what I mean?
It's like, it doesn't, I don't understand how they knew that there's 4 of these different.
Well, those they already knew that there were those, that when you looked at a DNA molecule, there are only 4 different molecules that contain nitrogen: adenine, cytosine, etc. Yeah, that they knew. So they had the puzzle pieces. They just didn't know which way the puzzle pieces went together. And I guess, and this is common in research, is they went to a machine shop, the guys at the machine shop, and they had them, start like creating metal bases so that they could put things together, and then try and fit it together. So it's really common in research, when you get into research, to have to build things that, at least in science research, that you have to build things that have never been made. So it's not only are you trying to do new research, but you're also having to sometimes build the equipment to do what you want to do that's never been done before. So that kind of adds another level of complexity when you're doing research, because you also have to kind of be able to kind of be innovative, right? In addition to thinking in a certain way, you also have to be able to figure, like, how can I make this particular thing function so I can find the information I want to find. When I was in grad school, I actually had to make circuits, and it was neuroscience, but I had to make electrical circuits so that I could measure this particular behavioral activity in rats. So I had to learn some electrical circuit building and do some soldering and all sorts of different things.
Wait, you couldn't have just outsourced that to like somebody?
Well, theoretically you could, but in actuality, researchers don't have that much money that they can just say, "Hey, I want you to build this for me."
Right.
And I was lucky because my professor was, his background was actually in electrical engineering.
Oh.
So he knew how to build circuits, and then he taught me how to do it. So I can do some basic stuff. I'm not very good at it. All right, so concept 16.6. Let me just see where this is going. Come on. Okay, so most of DNA, its job is to, wait, I'm seeing a different thing on my screen than you're seeing. That's weird. That's weird.
Because we have to end.
Oh my God.
Once again.
Checked out. Let me see if I can. I'm going to leave. I'm coming back. Promise. Network